Hello, my name is Professor Erik Murchie from the University of Nottingham. I'm pleased to be giving this talk called Canopy Environment Interaction in Crops, Light Dynamics and Photosynthesis for the International Workshop on Field Phenotyping and Modelling for Cultivation, JST. This talk follows on from Alexandra Burgess's talk on reconstruction techniques. A theme in this talk is going to be the link between canopy architectural complexity and what we call dynamic photosynthesis. So this is photosynthetic rate that's not the same, but it's changing. It's not static photosynthesis, it's photosynthesis that's responding to a change in environmental conditions, in this case, light intensity. So it's how we go from the complex canopy that we see on the right here to a complex photosynthetic response, where when we plot photosynthetic rate against light intensity, we don't get a straight red line, we get a scatter. Why does this happen, and can we use this to improve canopy photosynthesis in crops? I'm going to talk about three aspects of dynamic crop canopies. First of all, photoprotection with an introduction to photoprotection and how we can look at this at the canopy scale in 3 dimensions. And thirdly, I will look at the issue of wind-induced movement. If we make measurements of photosynthesis under high light and high temperature in the field, in this case this data is from Erie in the Philippines, you do not see a classic light response curve if you take random spot measurements of photosynthesis at ambient light intensity throughout the day. In fact, what you see is a scattering of photosynthesis. The red line in this figure shows an ideal light response curve with a high quantum yield and a high photosynthetic capacity. The real in situ photosynthetic rate is often lower than the ideal light response curve. Why would this be? Well, under high temperatures, high light and potentially high vapor pressure deficit, you see a reduction in quantum yield in the light limited phase. This can be due to photoinhibition or non-photochemical quenching, and a reduction in the light saturated phase and in the convexity can be due to rubisco activation, stomatal conductance, or severe photoinhibition. So many years ago we measured photoinhibition in tropical rice. Again, this was in Erie, in the field in the dry season on clear days, and we see a sustained reduction in dark adapted FV to FM, which lasts for several hours during the day. And this is a sign that chronic photoinhibition is occurring in these plants, and this is associated with a large increase in the deepoxidation state of the xanthophyll cycle, which is a photoprotective mechanism in all plants. So photoprotection can be defined as the prevention and avoidance of oxidative stress and photoinhibition, and has several components over different scales. So plants can change their morphology, so they can alter leaf angle, for example. You can have organelle movement, which is, for example, chloroplast movement, which can help to prevent overabsorption of light. Photosynthesis can acclimate and change the quantities of components of photosynthesis according to the long-term changes in light intensity. Non-photochemical quenching, which we will look at, is a photoprotective mechanism that is induced under high light. There are various electron sinks combined with PS2 damage and repair and radical scavenging, and all of this comprises a suite of photoprotective mechanisms in photosynthetic organisms. Non-photochemical quenching is a photoprotective mechanism that helps to prevent photoinhibition. But what is the impact of photoinhibition on leaves? Well, photoinhibition is defined as light-induced sustained quenching in photosystem 2, which can be due to damage. And This figure shows CO2 assimilation rate against light intensity. The first thing to note, one of the most common outcomes of photoinhibition is a reduction in the quantum yield, the light-limited part of photosynthesis. That's the light-limited range of light intensities. And as we go to high light intensities, photosynthetic capacity can be reduced in extreme circumstances. So a photoinhibited leaf that encounters low light will suffer from a reduction in photosynthetic rate. On the right-hand side, we can see that as we increase light intensity, chlorophyll excitation increases in a linear manner, and this occurs after the saturation of photosynthesis given by A max. This means there is always a surplus level of excitation energy at high light intensities, particularly for C3 species such as rice. So as we move from low light to high light, we shift from light harvesting to photoprotection. It's important to note that photoprotection, even though it reduces the likelihood of photoinhibition, it also causes a momentary decline in the quantum yield of photosynthesis. And photoprotection can last minutes or even hours depending on the prevailing conditions. On the left-hand side in this figure, you can see under low light there is a very, very efficient transfer of energy from the light harvesting complexes to photosystem 2 in this case, and hence electron transport. Okay, and under these conditions there will be no non-photochemical quenching, no photoprotection. Under high light, when light harvesting complexes receive more light energy than they can transfer to the reaction center, they will dissipate the excess excitation energy as non-photochemical quenching. This is a highly controlled mechanism that is regulated by the pH gradient, the proton gradient across the thylakoid membrane. And the operation of MPQ itself leads to a reduction in the quantum yield of photosynthesis. So you can think of it as a valve for excitation, excess excitation energy from the light harvesting complexes. So the outcome of this is that as a leaf moves from high light to low light, it will be operating NPQ, in this case is termed QE, and QE results in a lowering of the photosynthetic rate under light-limiting conditions. And a high-profile paper in Science by Kromdijk et al. Showed that by manipulating the relaxation of NPQ as it goes back from high NPQ levels to low NPQ levels under low light, affects CO2 assimilation rate in those conditions. And they were able to show that in the field, CO2 assimilation rate during high light to low light transients was enough to be linked to productivity over long timescales. So the response to dynamic light conditions seems to be suboptimal. This leads to a question about the three-dimensional properties of canopies, because the three-dimensional properties of canopies will change the light dynamics within those canopies. And so a complex environment for a leaf within a canopy, such as a rice canopy shown here, results in a complex pattern of downregulation of photosynthesis that affects biomass and yield. The question we then have is, what are the properties of canopies, the three-dimensional properties of canopies that can be changed to improve photosynthetic rate under these dynamic conditions? And this is where methods for reconstruction and understanding light dynamics from those reconstruction methods are important. And this is strongly linked to phenotyping techniques. I would like to first talk about manipulation of NPQ in rice leaves by overexpression of the gene PSBS. And we did this at first at the leaf level, and then we moved to the canopy level using 3D reconstruction. PSBS was discovered decades ago in Chris Nioggi's lab to be a regulator of NPQ. So the more PSBS you have, the higher the PSBS capacity and the faster NPQ is induced. We overexpressed PSBS in rice leaves and used RNAi technology to reduce the level of PSBS in rice leaves, and we were able to show this had minimal effect on the rate of photosynthesis of plants growing in a growth room. However, we did note in a glasshouse that when we tried to plot NPQ against CO2 assimilation rate, we noted that the higher the CO2 assimilation rate, the lower the NPQ value tended to be. To test the impact of the overexpression of PSBS on rice in the field, we could not do this in the field, so we used an agronomy glasshouse at the University of Nottingham, where we had supplementary light to increase the light level. We used high temperature, 30 degrees Celsius, and well-watered conditions. And the idea here was to measure the canopy level radiation use efficiency, the overall amount of biomass produced per unit intercepted radiation. The style of cultivation that we used in these glasshouses was as close as we could get to the field as possible. We were able to show in a paper in Communications Biology that the total dry weight per plant was higher in the overexpressors, and this was associated with a slightly higher increased leaf area, and we also noted a higher overall radiation use efficiency. So this is the amount of biomass that's corrected by the amount of intercepted radiation. We also noted a higher total dry weight and a higher filled grain dry weight. So our conclusion was that enhancing thylakoid photoprotection by overexpressing PSBS increases both yield and canopy radiation use efficiency in rice. So we noted that high levels of PSBS and NPQ do not have a significant effect under growth room conditions. When we move to fluctuating conditions and a canopy level environment, high levels of PSBS and NPQ do seem to enhance biomass in these conditions. We could not find a strong evidence of a mechanism like prevention of photoinhibition or the enhanced or reduced CO2 assimilation rate during the relaxation from NPQ. So we then turn to 3D canopy reconstruction to try to answer this. So what can 3D reconstruction and light distribution in canopies tell us about this problem? So we're moving now to photoprotection at the canopy scale in 3 dimensions. As you know, plant and crop canopies are complex systems. They are not just for photosynthesis, they are also for structure and reproduction and development. So for example, canopy photosynthesis might consider light distribution, light saturation, and be linked to nitrogen partitioning, but in the 3 and 4 dimensions we must also consider the timing of senescence. We would look at things like spike photosynthesis, the positioning of the spike and activity of spike in a wheat plant, for example. And this can also be linked to panicles of rice. We must also think about the fertility of the reproductive organisms. We must also think about the link with root traits, so lodging resistance and the adequate root activity for resource capture. So canopies, we may improve them for photosynthetic purposes, but we must also take into account the other functions of canopies. Is there a particular canopy structure that has an improved photosynthetic rate? Well, let's look at this. The figure on the left here shows a canopy with more upright leaves, and the figure on the right shows a canopy with flatter leaves. In the canopy with flatter leaves, much of the light is absorbed at the top of the canopy, This means that the leaves up at this part of the canopy are light saturated here, whereas leaves at the bottom of the canopy are very much light limited. So you have two extremes. If we take the canopy with more upright leaves, we find the better distribution of light, and the reflection of light down the upright leaf surfaces leads to much of the photosynthesis being close to saturation but not saturated. This has two effects. It means that there is an overall average higher photosynthetic rate in the canopy with upright leaves, but it also has a potentially higher optimal leaf area, because the upright canopy means you can have a higher leaf area index. So this affects both the distribution of photosynthetic activity and the distribution of resources like nitrogen. This has recently been shown empirically using wheat. So several years ago we developed a method for digital plant reconstruction, and this method was described by Alexandra Burgess in her talk that precedes mine, and she has used this method extensively and carried out many publications using this method. I refer you to her presentation for this, but just as a reminder, this method consists of 4 stages: image capture, point cloud reconstruction, surface estimation, and canopy formation. Plants are typically grown in the field or the glasshouses, and are then removed and brought in for rapid imaging, either manually or using a robot. The 3D point cloud is created. The surface estimation is carried out. This creates a 2-dimensional surface that we can use for ray tracing. And again, I refer you to Alexandra Burgess's presentation that precedes mine. So reconstruction and ray tracing is a powerful tool for predicting light patterns. So by capturing images and recreating a canopy using several representative plants from that reconstruction and surface estimation process, we can then apply ray tracing in order to predict light patterns within that canopy, and then we can link this to photosynthetic properties. Alexandra Burgess's talk will have described ray tracing, but just to recap here, in this method, and this uses the fast tracer produced by Jingguang Zhu and Xingfeng Song in their paper from 2013, and the tool they produced. So a known amount of light is applied to the top of the canopy reconstruction, and the path of this light is calculated through the canopy using rules governing scattering and transmission. For a given point in the canopy, fluctuations in light can then be calculated. So in theory, for any point in the canopy, we can predict the light dynamics during the day, and then we can also predict the photosynthetic responses during that day. So we can have a sum, if you like, of all of the leaf portions of a canopy. And we should be able to predict photosynthetic responses. This will take into account momentary shading, which will be due to the 3-dimensional properties of the plant. So this approach of 3D reconstruction followed by ray tracing has been used by a few groups, including us, to predict photosynthetic responses at the canopy level. So in 2015, we tried to use it to predict the impact of architecture on photoinhibition. In 2017, we looked at light partitioning in intercrop canopies, and in 2017, we also looked at optimization of acclimation at lower parts of a wheat canopy. And all of these approaches use this 3D reconstruction ray tracing approach. But I want to focus on photoprotection in this talk. So we asked the question, does photoprotective NPQ vary according to depth in a rice canopy? And we grew rice plants in our agronomy glasshouses, and we analyzed these at 3 different growth stages: growth stage 1, growth stage 2, and growth stage 3. These are all in the vegetative part of the rice life cycle. Growth stage 1 corresponds approximately to the onset of tillering, and growth stage 3 is just before anthesis. We used rice plants which overexpressed the gene PSBS to increase NPQ levels, and we also used the RNAi lines which had lower PSBS and lower NPQ levels in leaves. And the figure on the right hand side from left to right shows growth stage 1 to 3, and we can see it verifies the higher NPQ in the overexpressors compared to the wild type and the RNAi lines. We measured something called photoprotective NPQ. This is a specific measurement using chlorophyll fluorescence. It defines something called protective NPQ or PNPQ, which is the amount of NPQ required to prevent photoinactivation or photoinhibition. And it also defines light tolerance, which is the light intensity beyond which all reaction centers remain active or open. Why would we use this method? Well, it's a quantitative approach which defines the relationship between photoinhibition NPQ and the contribution to the decline in Phi-PS2. It's based on the premise that not all NPQ will actually be protective. Not all NPQ will actually prevent photoinhibition. It may be regulatory, or it may not simply be necessary. But even though it's not necessary, it may result in a reduction in CO2 assimilation rate. So it's a very useful measurement to make. So we measure this parameter called light tolerance, the light intensity at which 50% of reaction centers are photoinactivated. At growth stage 1, where the canopy is very open and there is less distinction between the upper and lower layers, the upper layer will have a higher light tolerance. As we move towards growth stage 3, which is a denser canopy, and there is a bigger difference between the light availability to the upper and the lower regions of the canopy, the light tolerance of the lower levels is much higher. So the photoprotective NPQ level of the lower regions of the canopy is actually much higher than the upper regions of the canopy. This comes as something of as a surprise. It is generally considered that leaves exposed to high light will have a higher light tolerance. This figure expresses that in a different way. We can see examples of light fluctuation within the canopy in upper and lower regions on the right hand side of this slide. On the left hand side of the slide, we see the percentage of the total leaf area that is photoinactivated against light intensity for the upper regions and the lower regions of the canopy. And it's quite notable that in the lower part of the canopy, a higher proportion of the canopy requires a higher light in order to photoinactivate PS2. So why and how is protective NPQ higher in the lower parts of the canopy? As I said, this came as something of a surprise. Well, it could be that light flecks or sunflecks presented greater economic risk via photoinhibition due to the lower photosynthetic capacity of the leaves. So they simply require greater protection from high light because the economic return of a photoinhibited leaf lower in the canopy is much lower. The mechanism for this is unclear, but we could look at things like PSBS levels and cyclic electron transport in rice leaves. The overall conclusion is that low light photosynthesis seems to be a very good route for improving photosynthesis, and we should look at the dynamics of photoprotection in these parts of the canopy. Lastly, I'd like to come to consider something novel, and that is the issue of wind-induced movement in canopies. When we consider an optimized canopy architecture, we look at properties such as architecture, so leaf number, shape, angle, plant height, etc. We can consider biochemistry, we can consider the activation state of rubisco, stomatal dynamics, NPQ, as we've seen in this presentation and acclimation. We can think of pigment composition and distribution. So there are some suggestions that having a lower pigment concentration at the top of the canopy may be beneficial to allow better light penetration, and things like nitrogen distribution. But there's one thing that's not considered very often, and that is this. So that is the very, very common wind-induced movement of canopies, and this will affect light dynamics. So wind, of course, has many effects on plants. So it affects plant development. It can induce dwarfing and changes in leaf size and shape. It affects the heat and mass transfer, gas transfer as well, boundary layer conductance. The canopy microclimate is greatly affected by wind, and there are influences obviously on damage at high wind speeds and rooting properties, and there's a big field of research in this area. But what's not been considered is the impact of low wind speeds on photosynthesis, small changes in canopy movement, and these happen very commonly. So what I'm going to talk about is low wind speeds, so not these high wind speeds which cause damage, Beaufort scale of 6 and above, but rather Beaufort scale between 1 and 3, so a light breeze. We believe this may be a new challenge for crop plants. Clearly wind will affect development of crop plants, but this has not been greatly explored beyond the effects, for example, due to lodging or damage to plants. And we may consider effects such as wind speed and direction, time of year, planting density and layout. All of these environmental considerations could change the relationship between wind speed, light dynamics and photosynthesis in the canopy. There is a precedent from the ecological literature. So an example is from the work of Piercy from the 1990s, who looked at the movement of leaves in trees such as aspen. And in these trees, they observed rapid leaf movement or twisting around the petiole. And this twisting around the petiole creates a rapid light dynamics for the leaves around it, and this rapid change in light intensity was shown to maintain, for example, rubisco activation state. So rapid leaf flutter caused by something known as torsional galloping could help to distribute light within plant canopies, and this is wind-induced. So in a review last year we proposed that the same principle could be applied to crops. We called this isolated leaf movement rather than flutter or torsional gallop, but it's the same principle. The movement at the top of the canopy can create opportunities for distributing light to leaves lower in the canopy. And modelling by Alexandra Burgess showed that a small distortion in a canopy, which corresponds to a light wind, this is in rice, produced a change in light distribution properties in the canopy. And we concluded that in fact the movement creates more opportunities for photon dispersal in canopies, more opportunities for photon penetration, and more opportunities for maintaining high photosynthetic rates. And we were able to show that the canopy photosynthetic rate would be enhanced via leaf movement in light wind. And we published this in Frontiers. So we proposed that of all of the factors that affect photosynthesis within canopies, affect photosynthesis within plant canopies, that is architecture, distribution of nitrogen, pigment composition and biochemistry, we believe that we should now be adding movement to this list of factors, because we believe that this does affect canopy photosynthesis. The problem then becomes how do you improve this in crops? The first step was to develop methods for tracking and quantifying movement of plants in the field as a result of wind. And we did this via machine learning techniques and tracking techniques. By training a network to recognize plant organs, in this case ears of wheat in a field, we were then able to track those ears in videos and then quantify the movement of those ears. And you can see here in this video the tips of the ears have been recognized and they can be tracked over time. And then we can then quantify the movement of each ear individually. So ideally we would want to do this with leaves, and this work is in development, but it's the first example of the quantification of wind-induced movement in crop plants, and it's an important first step. It's two-dimensional rather than three-dimensional at the moment. And I'd like to thank all of the people involved in this work. So also the funders, the BBSRC, IWIP has been involved, the Leverhulme Trust for Alexandra Burgess, and all of my group. Thank you.